Hi, my name is Di Bai. I'm a second-year graduate student from Rochester Institute of Technology, major in print media, School of Media Science. I've been working with Dr. Shu Chang and another graduate student called Venice, who is majoring in material science in RIT. Today, I'm going to give you a brief introduction of our research. Our research is mainly on visualizing and characterizing microstructures formed by printing particles using 3D imaging methodology. Additive manufacturing performed with solid particles can form microscopic structures based on the properties the particles used. These microstructures will impact the performance of the product. We have established the methodology to map powder particle positions in the interior three-dimensionally visualize the resulting particle structure. First, we make samples by using yellow toners or other kinds of printing particles. And then we use confocal laser scanning microscope in RIT to capture stacks of images. Under the microscope, the fluorescent printing particles are shown like this. What I'm going to do is to find the position and the radius for each particle. In this Leica microsystem software, people can take pictures of each layer. In each layer, you can see that the particles are cut into small green circles. As the layers go up, the circles become bigger or smaller, which means different position of each particle. From now, I'm going to show you two methodologies we used to obtain the coordinates for each particle. The first methodology was created by Venice. He used the Leica software to find the z position of each particle. As you can see the max positions of each particle in the statistic part. And then we took several pictures out and go to MATLAB to find the xy coordinates and the radius of each particle. The function he used is InFindCircles in MATLAB, and you can see more details on this website. The result of his methodology shows the xy coordinates and the radius of each particle. The second methodology, I generated a 3D Gaussian kernel to detect the xyz coordinates for each particle. At first, also from Leica software, exporting a stack of all the images into one folder. And in MATLAB, I created a short movie showing how the particles are shown in different layers. Then I used MATLAB to read all the images and build a 3D matrix with all the pixels values and all the images. Before convolution, I generated filters to get rid of noises and add a threshold to change the images to binary images. Finally, after the convolution between this 3D matrix and 3D Gaussian kernel, I got another 3D matrix which is called C-image. Then by giving the image area, as you can see here, I input the XYZ dimensions range. And the results are shown in row, column, and V. V is standing for the Z position. In order to compare my results with Venice's results, I plot a 3D image with all the centers and the particles as you can see here. The blue points are my centers, and the red points are Venice's centers. They are pretty close. I even make a two-sample t-test, and the result proves that they are close to each other. So after introducing these two methodologies, now I'm going to show you how to reconstruct the 3D microstructure of the toner particles. First, I use scanning electron microscope to capture the images of the same toner particles we used under confocal microscope. Since SEM is based on the surface visualization, it cannot visualize the interior structure of these particles. But SEM could still help us to correct the radius of the particles taken by confocal microscope. We took several images under electron microscope, and here's one example. I used ImageJ software to analyze the radius of the particles. As you can see here, first I give it a threshold to change the images to binary images. Then after setting scale, the dimensions of this image is changed from pixel by pixel to micron by micron. Then I choose area and Ferris diameter under the set measurement dialog. Ferris diameter can help us to calculate the maximum and minimum diameter of the ellipse detected by ImageJ. Then analyze particles. The results show that ferrite is standing for the maximum diameter and the minimum ferrite means the minimum diameter. After analyzing SEM particles, I used Minitab software to create a histogram of the particle's radius under confocal microscope and scanning electron microscope. As you can see here, the mean values of these two histograms are pretty close. Eventually, I reconstruct 3D structure of the toner particles in Reno. The middle one with XYZ coordinates shows the positions and the radius of each particle within specific image areas. In this video, the box outside the spheres is the image area. And I calculated the packing density of this sample. Packing density is the total volume of the spheres divided by the volume of the box taken by these spheres. On the right, the top one shows how did I calculated the coordinate number, which is the number of the spheres in contact with any given sphere. On the right, the bottom one, it shows how did I calculate the void areas. As you can see, the red spheres are the void areas. We are still working on other microstructure parameters like the nearest neighbor distance and the contact angle. That's pretty much about what I have done in this research. Thanks for the help from Dr. Shu Chang, Dr. Howard Mises, Professor Marcos Eastman, and Vinis Patil. My name is Bai. If you have any comments, questions, please be free to send me an email. Thank you for watching.